going to be talking today about a package I've been working on called Celaref for annotating single-cell RNA-seq clusters by their similarity to a reference dataset, not actually doing any clustering. We asked people to do it beforehand as well. Yes, and I have to thank Liam for introducing all the single-cell kind of concepts because it makes it easier. So obviously, you take a tissue, any particular biological tissue sample, and there's going to be a huge mix of heterogeneous cell types in there. With the, up to a few years ago, if you were interested in looking at the gene expression, you kind of had to look at the mix as a whole. But now we've got the single cell technologies just in the last couple of years. It's possible to single out the cell type that you might be interested in and sort of ask questions specifically for that. And I'll just give another overview of the workflow I'm going to be focusing around, which is when, if you start with a heterogeneous single cell sample, you then go and you pick your single cell sequencing technology. There's a whole bunch of them out there, but you just pick one which suits your needs, and in any case you're going to end up with your sequencing data out at the end of it. We take those sequences, we align them to a reference genome, and to generate this counts table. And all that is a big table with cells 1 to 2,000 odd, however many you have, and for each cell, how many times each gene was seen in that sample. So 0, 1, 5, however many times. And that's a really sort of core result in any sort of RNA-seq, and especially a single-cell RNA-seq analyses, as you've already seen them before. We don't know anything about those cells at this stage. We just have, they've just been numbered 1 to whatever as they've come out off the machine. So to actually do anything useful with them, it's one of the really common steps is to, just say, look, let's group them together, and we look at, we can tell, we want to analyse the ones that are similar to each other and say that that's a cell type. As controversial as that term can be sometimes. We know the blue ones are similar to the blue ones and the yellow is yellow, but we don't actually know what those are. So touched on, there are a lot of different clustering tools out there. It's a really gnarly, computationally heavy sort of bioinformatics problem. There's a lot of tools. I'm not going to talk about them because I'm more interested in what you do next. Because if you have, what cells are like each other, but you don't know what type of cell that is in a sort of biological sense. And this actually came out of sitting around in a meeting with looking at a new single cell data set, going, this really interesting. Where are the oocytes cells? Like, is it this group? What's that group? Oh, that's immune cells, don't care about those. Just trying to figure out what's what, and, so that you can then get on to the next stage, which is looking at the biological question. So there are obviously a bunch of ways to annotate your single cell clusters, and that first one, don't. Actually go back and use a different clustering tool. There are a few that are aware of, can be aware of different tissue, sample types and reference data. Of course, you can definitely do this manually. Get someone who either know about your cells that you're looking at, or find someone who does, a real biologist. Sit down, look at the cells, see, okay, what marker genes can we see? What's expressed here? Okay, that's a neuron. That's got this expressed. Obviously, really sort of good results there, but you need to have an expert, and it's very time-consuming. Which brings us to the third option, of which Celaref falls into, which is a cell cluster labelling tool. So there's a few of these around. Celaref is a little bit different in that it works at the group level. Other tools that I've looked at have worked at the a cell at a time kind of level, and I'll come back to that later. So this is Celaref. It's up on GitHub if you're interested in looking it up. But what it does, its core function is basically to, take a single cell sequencing experiment results and a reference data set from a similar tissue or similar samples and as a reference, and then take use information from that reference and try and make up labels for your cluster, for your new clusters based on, hey, what group does this match? Basically saying, just picks matches on the basis of some references. I'll get back to, but it makes an attempt to do this at the group level, the cluster group level, and quantify that by a p-value, all based on differential expression, because that was easy. Here's the input and the output, basically. So to use this package, you need to have, for your experiment that you're interested in characterising, you need to have that counts table I mentioned at the start, and you also need to have those pre-computed cell clusters, which you did on somebody else's server. And then for the reference data set, you need to have exactly the same pieces of information, but you just also need to know, hey, what are those cells, because else it's not much of a reference. And from there, a tiny bit of code for example's sake, but you get out suggest some cluster names, whether there's no similarity or one or two matches or more than that, and a p-value, it's some sort of measure of the confidence in that assignment, and a visualization, which I'll go through and explain, but just to, for looking at those corner cases where you're not sure what's going on, just something to figure it out. Okay. So I said this is based on a differential expression analysis. So what I mean by that is for every cell cluster that I'm, characterising, we'll start with, pulling out what's different for this particular cluster versus everything else within that same experiment. So, we're forgetting about the reference at the moment. We're just looking at our query experiment and seeing, okay, well, what genes are particularly high or low in this particular cluster. And the idea of doing that is just a really sort of intuitive approach to say, okay, well, if we see a group of genes that are particularly highly expressed in our query in this, purple cluster, and we see that same or similar group of genes highly expressed in in the neuron cluster of our reference data set, we say, okay, well, maybe that's a match. And that's basically the approach here. But this being biology, you can't just pick two gene lists and overlap them in a big Venn diagram and expect to get anything useful out of it, because it's never that clean. So hence we use the differential expression. So taken a sort of full gene set ranking kind of approach, where we calculate the relative difference of each gene's expression. Can you see my pointer there? Yes, you can. And then rank them for the most enriched to the least enriched, just based on a fold change, a confidence interval. We do it that way just as a compromise between the magnitude of the change and the statistical confidence in that change. But at the end of the day, we have a rank of 1 to 20,000 genes, or however many you have. And we do that for our query experiment. We also do the same thing for the reference experiment, so this pre-processing stage is just the same. But what we then do with the query class that we're interested in is pull out this list of top genes, which are just those that are statistically significantly overrepresented in the query group. And we take those top genes that we think, yep, they're representative of this query cluster that we don't know what's in there, and we look up where they fall in terms of the entire rankings that we also, the full rankings that we computed for the reference clusters as well. So these 6 columns here are 6 different reference groups that we already know what they are. And these little black tick marks here are just our top genes from the query cluster. And the big black bar in the middle is just the median rank for ease of visualization there. And you can see here that we're just plotting the same set of genes on each of these different reference rankings. And we want to see the one up the end here in the pink, where those top genes from the query also happen to be the top genes in this particular reference group. So we suspect that, okay, well, what's this? These are oligodendrocytes. And those are actually oligodendrocytes because that's real data. I'm been testing this with this example is from a query, this is human brain data set versus a mouse brain data set. Here's the full example. Obviously you do all the clusters at once, because why not? And output of the Celaref here would be this plot and a label generated for each of the each of the clusters, and a sort of a measure of the confidence in that. Okay, and I'll go through some examples. Okay, so this first example here is probably the easy case. So this is obviously these are two perfectly well characterised data sets, so we know the input query that we don't know is actually astrocytes. And you can see here, so I've got the top genes for the astrocytes plotted against the six reference genes, reference groups. And you can see up here, there's a clear forerunner here. It's got the highest median gene rank. But rather than doing all versus all, I want to quantify how much higher that is to the rest of the groups, because we're looking for the big sort of step down here where there's a difference between that group and everything else. So we do that via I rank every reference group versus on their median gene rank, and then compare the distributions, pairs of distributions with a Mann-Whitney U-test. And in this case, obviously, that first example is going to be very significant, and that 1 to 2. So yeah, that's the p-value that we reported previously, and we then pull out what that cell type is from the reference, and that's astrocyte epidermal, and that's how we form that cell label, that group label. Sorry. In the second case, you can see it's a lot less, clear-cut. There's no actual forerunner here. But we start doing the test in the same way. So look at, this one's the top. It's not very high, but it's the top ranked median gene rank. Compare that to the second one. Obviously, there's no significant difference there. So I then compare the second to the third. No significant difference. Third to fourth. You just keep stepping down until we get to the end. No significant change in the distributions anywhere. So we report, hey, there's no similarity here. And that's what we'd actually expect to see for this example, because if you dig into the this paper that we got the data from, they actually call it the hybrid group, and they describe that as the cells that they looked at and couldn't put into a particular category. So it's the other cluster. So it was good. The last case here is an interesting case. So you can see, if you just look at this plot, there's two very high distributions here. And if we actually do the same thing, we start comparing the top one to the second top one, it's not actually any different in terms of distribution. So we keep going and then compare the second top to the third top, so that's the green to the yellow. And of course that is actually a significant difference in the distribution, the Mann-Whitney U-test. So we report that p-value and then take both of those first two groups. So we say that those are pyramidal SS and interneurons for the label, and we need to be able to do that, have that sort of one-to-many mapping, because in what happened here is that in the brain data set they've got one cluster of cells which are called neurons. But in the mouse data set, they've subdivided their neurons into these pyramidal SS neurons and interneurons, and we've been able to sort of detect that both of them have been smushed together in the labelling there. And there's also a few other heuristic kind of information that we apply to these labels as well. We check it's above random, and also report any reciprocal hits. I won't go into that now, but it's there. Okay, so this is, I want to do some wider testing to make sure we're not just going to label everything in sight with everything else in sight. So here is, again, these two brain data sets in the blues, and the green data set, or the green data sets, another experiment which is derived from a mouse lacrimal gland, which is like a gland, I think, that feeds the tear ducts. And so you really don't expect there to be that many cells that are in common between the brain and a gland. And as you can see here, Mark did an all versus all comparison, and highlighted the matches that contribute to labelling here. And you can see there's these cliques with the astrocytes. You've got an epithelial group over here, neurons with the 2-to-1 mapping. And then, so that's all very nice. You've also got things like this, which has one of each. And that actually is, those are microglias and macrophage. So immune cells, and biologically speaking, they're actually quite similar. They're based in different tissues, so that's really nice to see, that sort of thing. And you get something similar happening here with the endothelial cells. A few other matches around, but obviously the thickness, of course, is representing this sort of the negative log p-value of that match, so confidence of that. Okay, so I've also tried this on PBMCs from blood cells. I won't go through these results here, but just suffice to say I've tried it. Tried it because PBMCs are actually much harder to do because they're quite similar cells than, compared to different tissue cells. Okay, so I'll come back to this comparison with the other tools. So this is this big, nice looking Sankey plot here is from SEMap. And so SEMap works, it does a per cell assignment from, a query group, a query data set to your reference data set. So here's the 2 brain data sets again. And, so you end up with sort of a proportion of how many cells from group got assigned to your other group. Whereas in Celaref, you get it makes an attempt to generate these labels and gives you a p-value there. But, despite this very, very different statistical approach, but you actually do get some reasonable, good agreement between the 2 tools, which is good. Okay. So just to sum up, you can use the Celaref package to try and make labels if you've got an uncharacterized new, single cell sequencing data set. You don't have to have a perfect reference, if there's a few differences. I've actually tried it with some, that PBMC reference is actually microarray data. And it's also tractable, I think, using the differential expression approach because, at the end of the analysis, you can then go and pull out the differential expression analysis and look up the genes there and, that's the most computational heavy step and you can reuse it. And, I think one thing that might also be interesting to this audience is that it could also be used for looking at evaluating different sets of clustering on a particular data set, which I believe is something that Luke is going to expand on a lot more and in his talk next. So obviously this is on GitHub. It's not in Bioconductor yet, but that's coming. And there's a vignette here with a lot of the example data and how to generate it that I've talked about here. References. And lastly, I'd just like to thank everybody from the Monash Bioinformatics platform. That's us in bitterly cold Melbourne. Dave is our group leader. Sonica is our manager, who's been very involved in this work. And Paul has been very patient with my various statistical questions. Thank you.
Any questions? I would just want to ask, because here you only have 6 cell types, but how about if immune cells, you probably will have 30 or 40. Do you consider they're multi-paradigm correction FDR values instead of the p-values?
So the question was how this will perform with lots more reference cell types, like you might see in an immune experiment. Yes, I think it will perform okay. So I've done some simulation data on anything from 3 to 500 different reference groups. It actually gets statistically more difficult to assign a group with higher numbers of groups, so I haven't done the FDR in that direction. But yeah, it seems to work.
The p-values will increase as you number of clusters.
Yeah, yeah, because it's looking, it becomes harder, yes, I think.
Okay, please join me in thanking Sarah for that.